Thank you. I'd like first to thank OMICS group and my colleague of the organizing committee for the wonderful collaboration. The title of my presentation today is about the phytotherapeutic potential of selected Middle East natural products. The outline of presentation will be focused on 4 main points: introduction and objectives, experimental procedures, results and discussion, conclusion, and future work. As all of us know, natural products can be derived from different sources, either plant, animal, microbial, venoms and toxins, or marine world. Among the aim of Pharmaceutical Sciences Department, which I have the honor to be the head of this department, is the development of research skills in under and postgraduate students. The team members actually were working together to fulfill this objective. Overview of Pharmaceutical Sciences Department. Actually, the department has 3 main specialties: pharmaceutical microbiology, pharmacognosy and medicinal plants, including phytotherapy and phytochemistry, organic and medicinal chemistry. The statistics previous studies revealed that the Pharmaceutical Sciences Department is among the productive departments at the faculty and university level regarding research activity. The main research areas actually are diverse to fulfill achieving the mission of the department and its objectives. However, I am going to highlight the specialties relating to phytochemistry and drug discovery of natural origin, in addition to the medicinal chemistry and drug discovery of synthetic origin, since both of them, of course, are the backbone for pharmacy profession and pharmacy industry. At the same time, we are concerned with not only with drug discovery, but also in collaboration with the Department of Pharmaceutical Technology at the same faculty in order to have a role in pharmaceutical drug development. In doing so also, we create this culture among our students in the undergraduate level. We have 2 junior research teams in the field of phytochemistry, phytotherapy, in addition in the field of medicinal chemistry. Last year, the students, with their executive supervisor, Dr. Karim Rafat, managed in traveling to Germany to see updates in research in honorable universities and pharmaceutical companies. And fortunately, this year, next week, they are going also to Germany to complete their scientific activities. Now, moving back to the title of my presentation about potential therapeutic effect of selected medicinal plants of the Middle East region. Actually, these are the selected plants presented here, coming mainly from Egypt, Syria, and Lebanon. We are concentrating actually on 2 main areas that are very important for us regarding the glycine receptors and how to discover drugs of natural origin to protect the glycine receptors. And of course, as some of us heard about that, and they have, of course, an idea about the glycine receptor. So I'm not going in details about that, but just I would like to highlight this area of research, which is very interesting in our department. And we refer also during carrying out this study to strychnine, as of course all of you know that it potentially blocks the inhibitory actions of glycine. And actually, phytochemicals can play a role in this part, as I will demonstrate. Usually, we consider in our studies the animal behavioral test, convulsion and death test. This is very important. Not only we rely our results only in vitro studies. So phytochemicals which reverse the action of strychnine have been shown to have anxiolytic properties. So, actually, regarding the phytochemical scheme for separation, we collected these plants, and the plants are, of course, subjected to extraction and fractionation using different procedures. After that, the extracts of the plants were examined and tested regarding different pharmacological activities, mainly on diabetes, for protection from diabetes, and also diabetic neuropathy, which is usually the complication of diabetes, and can be measured by hot plate flick test and tail latency test. At the same time, the reduction in blood sugar level was observed after measuring in the blood sugar level in vivo. In addition, the antioxidant activities of the chosen plants were also assessed in vivo. Regarding the plant Rhium ribus rhizome, this plant is available at the herbalist in Lebanon and usually bought by a lot of people in order to, of course, sometimes people believe that they could manage their blood sugar level when they use this plant. So this plant also was subjected, and this plant is very important in Lebanon, was subjected to extraction and diffractionation procedure, and the same tests were applied as the previous plants on this plant. Regarding the protective effect of Oregano marjorana, which is a very well-known plant present in Egypt and in Lebanon, and in a comparative study between these two plants, it was found that they have a very promising neuroprotective activity of glycine receptors. And actually, the HPLC studies revealed that the presence of phenolic compounds in both plants, of course, appreciate this activity. The studies also revealed that the amount of phenolic compounds are present in the Egyptian species rather than Lebanese species. However, both species could be promising drugs in protection of glycine receptors. As we can see here also, the dose to exert its effect, the IC50, is always low in the Lebanese plants. And of course, it seems to be that both plants can serve as new sources of drugs in this concern, especially both Lebanese and the Egyptian species. Another plant, which is a very important plant, indigenous only in Lebanon, endemic to Lebanon. And this plant is Salvia Lebanon-Chica fruticosa roots, and the study of the blood sugar level was carried out, an acute effect on the blood glucose level. And as we can see, there is a marked decrease in blood sugar level, of course, after induction of diabetes and using a glibeclamide as a reference. This also, in vivo study, again, subacute effect on blood glucose level of the same plant, the roots especially of the same plant. Why the roots? Why the roots was chosen in this study? Because the roots was found to be rich in diterpenones, which is a very important class supporting the antioxidant effect of these plants. Mainly the antioxidant effects of the medicinal plants actually due to either the presence of phenolic compounds, polyaromatic compounds, flavonoids, and anthracenones. And here, Salvia Lebanon-Chica fruticosa is an endemic plant to Lebanon, was found to have different activities regarding a marked reduction in blood sugar level compared to glibeclamide, in addition to diabetic neuropathy management and the thermal latency, which is among the most important diagnostic complications of diabetes. The acute effect also on blood glucose level was found on Centaurea horrida because it is purchased at the herbalist, both herbs and roots without discrimination. That is why we studied this plant as it is used by the public. And the same again, we can find that the acute effect appear on blood glucose level and also on subacute level. And in fact, the activity is proportional to the concentration of the drug, so it is dose dependent. In vivo assessment of the antioxidant activity was also in the favor of the activity of this plant, of course, by measuring a catalase level. So all these studies were in supporting the possible use of this plant for its anti-diabetic effect. Furthermore, the diabetic neuropathy also can be overcome also by using of the plants with the same form as used by the public, both herbs and roots. This is also another plant called Horridium sponatum, and this also has acute effect on blood glucose level as illustrated here in this table, and at the same time at the subacute level. And the in vivo assessment of antioxidant activity was also in favor of this plant due to the accumulation of polyphenolic compounds in this plant according to literature studies. Diabetic neuropathy management and thermal latency was measured also, and it was found that it is effective in this concern. Rhium ribus, which is a very important plant because it is available in the Lebanese market. Also, the rhizome is available there in the Lebanese market, and it is very important to mention that the effect was found to be A normal normal glucose level, it is okay, and just the decrease in blood sugar level in normalized rats is cannot be measurable. So it is effective even to adjust the normal blood sugar level. In addition, of course, to its effect in general to decrease the high blood sugar level. So this plant, I'd like to focus on the importance and significance of this plant because it acts both on normal blood sugar level and also in elevating or high blood sugar level. So this is also the studies here. We have made a lot of studies about this plant, and fortunately we found that all the studies revealed that there is a decrease in blood sugar level in both normal conditions and in high blood sugar level. The same also in regarding diabetic neuropathy. So in this case, when we investigated by HPLC and comparison with reference samples, the retention times, we found that the plants accumulate high percentage of rutin, which is a very well-known flavonoid. In addition, the rutin-rich fraction was also active. However, the total extract was found to be much more active than the other two samples. So when we examining this plant, we found that in addition to the presence of flavonoids, which constitute rutin about 31%, we found also the presence of anthraquinone like homogene and chrysaldine. That's why we can also correlate the phytopharmacological effect, of course, based on the accumulation of anthraquinones besides the already present and detected flavonoids. Regarding the integrated approach with pharmaceutical technology, actually this is the most important aim also of our research activity, and we have managed to do that by a collaboration work between my colleague at the Department of Pharmaceutical Sciences and my other colleague in Department of Pharmaceutical Technology. He was at that time present in Beirut, Faculty of Pharmacy, Beirut Arab University, and now come back to University of Alexandria, Department of Industrial Pharmacy. And they managed together the work, of course, attributed to both of them. I'm not involved in this work, but also it is among the departmental images that I want to highlight on. The amelioration of diabetes and painful diabetic neuropathy by Punica granatum extract and its spray-dried biopolymeric dispersions. And as we can see here, my colleague prepared the two formula with casein and the other formula with chitosan, and they compared the activity after, of course, regarding diabetic neuropathy and regarding also the blood sugar level, and it was found that the formula 1 can be used in fast-release medication, and the formula 2 can be applied if we need to have a sustained-release medication. And this also, I'd like to mention the name of my colleague, Karim Rafat from the Department of Pharmaceutical Sciences, and Wael Sami from Department of Pharmaceutical Technology, Beirut Arab University, Faculty of Pharmacy, and now back to Alexandria University. So this is the results, and in general, I will just here display some of the publications regarding the research activities of the department. As you can see here, and in endogenous Lebanese plants treating diabetes and related complications, with sometimes plants coming from Syria and be, of course, purchased in Lebanon. And also we have investigated the Lebanese species, Syrian species, and at the same time the Egyptian species. So as we can see from these publications that a lot of medicinal values can be attributed to plants of our area. The need only to highlight on their importance and to apply techniques for drug delivery that will help and accelerate, of course, and secure our people, since we have our own treasures. Before ending my presentation, I'd like to acknowledge Dean of Faculty of Pharmacy, my colleague, Professor Dr. Abdallah Laani, and Dr. Karim Rafat, my colleague, of course, for the our co-authors together. At the same time, I'd like to convey my thanks to the genuine research teams because they helped us in doing some research activities. And thank you for your attention.
Thank you, Maha. Any question?
What is the mechanism of action of the drug in the diabetes, either by insulin increase or by increasing the sensitivity or by decreasing the glucogenesis in the liver? I mean, what type of mechanism it follows? Second question is
Yes, I will tell you, actually, it depends. The activity mainly depends on the antioxidant effect produced by the natural products, especially the structure activity relationship need to be an antioxidant is to be, of course, aromatic and polyphenolic to, of course, compact any, free radical and capture free radical and prevent the invasion of free radical to the intact cells. The studies activity actually depends on, first of all, on folklore use of the people, because they have this culture and they have this belief and concept that these plants can protect them from diabetes or either can help them in treating diabetes. So what we carry out is to have evidence-based medicine regarding the use of these plants in treating diabetes. At the same time, of course, you know that the antioxidant effect can be measured by many ways. For example, by measuring the level of catalase enzymes, so it is an indicator also. Measuring some, of course, enzymes, sometimes it happens, for example, in liver. We also measure the oxidative enzymes, and we, of course, the level of the excretion level of the enzyme will be affected, of course, by the presence of these antioxidants. So actually, the main activity is based on the antioxidant activity of these compounds, polyphenolic compounds, aromatic, flavonoids, and anthracenones, which is actually, we found that a collection of these categories is present among the chosen plants. At the same time, we can find the percentage of routine, 31%, one of the very important flavonoid act as antioxidant in Rheum ribus, which is very important plant really need to be considered regarding future perspectives.